In this tutorial, we are going to continue with the regulation of gene expression. We've already done the introduction. We defined what gene regulation is. We said gene regulation is just the process of increasing or maybe decreasing the production of proteins, and we defined what gene expression is. We said it is just the process by which the information which is in a gene leads to the production of proteins. We defined what now gene regulation or regulation of gene expression is. So we said that regulation of gene expression is just now the steps or the processes which are involved to increase or decrease the production of a specific protein. We also looked at the importance of gene regulation. We said that it saves energy, and other than that, it ensures that the proteins which are made, they are made when they are needed. They are made at the right time and where they are needed. They are made at a specific location or a location which is in deficiency or in need of those proteins. We went further to look at the two classes of genes. We said there are structural genes. These genes are the actual genes which code for the proteins or which forms proteins. Other than that, we said there are also what are known as regulated genes. The regulated genes are the ones which control now the protein synthesis. And we said that regulated genes are a lot. They are about 98% of the genes, and the structural genes are just 2% of the total genes. Other than that, went further to look at the structure of a certain gene. We said first, a gene is a portion of DNA that controls a specific characteristic. We looked at the structure. We said that since this gene is made up of the regulatory gene and also the structural gene, we said that there is also a small portion which we call a coding gene. Now, just few bases behind the coding gene, we said there are what are known as the regulatory sequence. The regulatory sequence are the ones which are able now to combine with the regulatory gene in order to form what are known as or what is called the regulatory DNA. We went further to look at the operation of the RNA polymerase. We said it's bind to the regulatory sequence. After it binds, it then moves and transcribes the genes which has to be transcribed. So this is the summary of what we looked at in the previous session. If you have not yet understood, or if you didn't look for a video on regulation of gene expression, or if you haven't covered it yet, you can just search on the channel, just search for regulation of gene expression, Meticulous Hub. The video will come so that you understand the first part and we move on the same page, because in this discussion our main focus will be on the Lac Operon. So let's now dive into this and understand what the Lac Operon is. So what is the Lac Operon? So when you're talking about the Lac Operon, so this is what we looked at in the previous sessions, and also this is also what we looked at. So this is where we ended. We ended here on the RNA polymerase after binding to increase or decrease the forming or conversion of mRNA and the other things which we discussed. Okay, so proceed from here. Now let's understand this Lac Operon. So when you're talking about the Lac Operon, this lac comes from the word lactose. So even as we are discussing the Lac Operon, we need to bear in mind that we are just talking about this lactose, how this lactose is able to be utilized in the absence of glucose. Okay, and we said that this lactose is just a disaccharide which is made up of glucose and also galactose. So when glucose combines with galactose, it forms what is known as the lactose. So when you're talking about the Lac Operon, you're just talking about the group of genes that regulate the metabolism of what? Lactose. The group of genes that regulate the production or the metabolism or the breakdown of this lactose to form glucose and galactose. Now let's dive into it. So we have a certain bacteria which is known as the E. coli bacteria. So this E. coli bacteria is found in our bodies, okay? Specifically, it is found in what is known as the GIT, the gastrointestinal tract. This E. coli does something to our bodies. It aids or helps us in the production of some certain, or in the breakdown rather, in digestion of some certain food nutrients. And in return, what does it retain? Or what does it gain rather in return? It gains shelter, it gains transportation. So there is that symbiotic relationship, that mutualism that exists between the organism where this E. coli is residing and also the E. coli and that certain environment. So there is what is known as the mutualism, or what is known as the symbiotic relationship. So this E. coli is able to obtain shelter from us, okay, human beings. And also this E. coli gains, apart from that, it gains food from us, the nutrient, okay. The shelter, the transport, it is from us. Then this E. coli, it also aids us now in the breakdown, in digestion, okay? So this is what we gain from E. coli. So it is a 50-50 gain. It's not like this gains more, then this other one gains less. No, it's 50-50. So this one is depending on on this organism. We are depending on E. coli for digestion or to aid in the digestion system. And also E. coli is depending on us for shelter, food, and transport. So this is what we need to understand, okay? And since it is depending on our food, meaning that it uses this glucose as the source of energy, okay? It uses this glucose as the source of energy. Now, where is this lactose found? When you check on the milk content contains this lactose, okay? No wonder milk sugar is what is known as what? Lactose. The other name for lactose is milk sugar. So milk sugar is simply the lactose. Now let's dive into this Lac Operon. So what is the importance of this Lac Operon? What is the importance? So let's look at the importance of this Lac Operon. So this Lac Operon helps in the breakdown, as we have said. So it helps in the breakdown of lactose, helps in breakdown of lactose to what? To glucose and also galactose. So that is the function of this Lac Operon. So this bacteria, we've said the E. coli. E. coli, there are certain bacteria which just lives in our bodies. Those bacteria are known as the normal flora, okay? E.g. the E. coli. These bacteria do not cause infection to our bodies. The body depends on that bacteria for a certain function, e.g. as we've discussed, in digestion process, okay? Now let's try to understand the structure of, or the operation of this Lac Operon. So I'll have this, let's say, in the previous class we said we have what is known as the regulatory gene. The gene is divided into two. So this one is what we are calling the regulatory gene, okay, or the regulatory portion rather. Then we have another portion. Let's say we have this, what we call the structural gene now, okay. We have this portion known as the structural gene, or the structural portion rather. Let's call it the structural portion. So this structural portion has certain composition, okay. Let's say number one here, it has what is known as the lac Z, it has that what is known as the lac Y, it has also what is called, so this is lac Y, it has also what is known as the lac A, okay. Then of course after this structural gene we'll have a terminator there, okay, to end the process. We have a terminator there, and this lac Y, lac Y, lac Z, and also lac A, they have different functions, okay. They have various functions. Let's look at the functions of this lac Y, lac Z, and also lac A. Let's look at their function. So what we need to understand is this. When you're talking about the lac Z, so this lac Z produces some certain enzyme, okay. It's produced the enzyme which is known as beta galactosidase, okay. So this is the enzyme it produces. So this lac Z produces what is known as beta galactosidase. Now, what's the function of this beta galactosidase? So this beta galactosidase is the one now which is involved in the breakdown of this lactose, okay. It is involved in the breakdown of this lactose to form glucose and also galactose. Now, what about lac Y? So lac Y, this segment also produces the enzyme. The enzyme which is produced is known as galactoside what? Permase. Just from the name, permase. We are looking at galactoside permase. So just from the name, permase, permeable, right? So this one makes the cell membrane permeable, so that the lactose can now be pumped into the cell, okay? So makes cell membrane permeable, so that to allow pumping of the lactose into the cell, okay. So that is basically the function. What about the lac A? So lac A, the lac Z, we've said that it produces what is known as the galactosidase, right? The beta galactosidase, which aids in the breakdown of lactose into galactose and also glucose. The lac Y, we've said it produces a certain enzyme which is known as galactoside permase. The galactoside permase is the one which makes the cell membrane permeable, so that more lactose rather can be drawn into the cell. Now, this lac A, it also produces an enzyme which is known as the beta galactoside So this is the beta galactoside transacetylase. Okay. Now, this is not much of our importance. The beta galactoside transacetylase is not much of our importance at this stage. What we need just to concentrate on is the lac Z, and also the lac Y. Okay? So this one codes for the beta galactoside transacetylase, which we'll understand later on. Okay, this is not much of our importance at this step. So let's look at this now. So we've said we have the two portions. The Lac Operon is divided in these two portions, which we are calling the regulatory portion, okay, or DNA rather, and also what we are calling the structural portion. So one thing I want us to appreciate here is that the structural portion or the structural gene is the one which forms those enzymes, okay? It is the one which is able to form the enzymes which are involved in gene expression. So that is very much important for us to understand. Now, the regulatory gene can be switched on. It can be switched on sometimes, and also it can be switched off. So when is this regulatory gene switched on? For example, when the protein synthesis has to take place, this regulatory gene has to be switched on, okay? But when there's no protein synthesis rather, this regulatory gene does not need to be switched on because it won't perform any function, okay? This won't perform any function, so there's no need to switch it on. Now, I want us to understand the regulatory gene. We've looked at the structural gene. Now, what about the regulatory gene? What is it about? So, this regulatory gene is made up of different portions, okay? Or this regulatory portion rather. It is made up of different segments. The first thing, it is made up of the lac I. I just means inducible, okay? The lac I. So lac I produces a certain enzyme, or it codes rather for a certain enzyme, which we call the repressor. So this one codes for a certain enzyme known as the repressor. Then the regulatory portion rather, it's made up of the operator also. Other than the operator, it is made up of the promoter. Other than a promoter, it is also made up of the terminator. Even though this terminator is found just at the end, okay? So even if we don't include it at this stage, but we'll look at the terminator also. But let's understand that it's made up of the lac I, which produces a certain enzyme, the repressor, the operator, and also the promoter. Now, let's understand something interesting here. If we have, so this is the strand we have, okay? The genes we have here. This side we have the regulatory portion. This side we have the structural genes or the structural portion, okay? So here we have let's say we have this. So we have the lac Z there, we have the lac Y, we have the lac A, okay? So this is what we call, or what we are calling rather, the structural portion. Then this side we have the regulatory portion. Okay, so the regulatory portion, we've said that it is made up of the operator. So let's have an operator there. So this is the operator. Then other than the operator, it is made up of the promoter. So let's say we have the promoter there. So this is the promoter. So behind the promoter, we have the lac I. So this is where we have the lac I there. So lac I is there. So now let's understand what really happens. So when you're talking about the promoter, this is where the RNA polymerase is going to bind. Okay? So the RNA polymerase binds the RNA polymerase. It binds to this promoter. Okay? It is bound to this promoter. So it binds to the promoter. So we need to understand that. So this RNA polymerase, of course, we have just behind the promoter, we have this cap. We'll look at the function. Okay? So this RNA polymerase binds to what? To the promoter. Now, it can transcribe the genes we have in this structural portion. It can transcribe because there is this operator, there is this portion known as the operator. So let me get rid of this. So there is this portion known as the operator. So this operator, what is the function of the operator? So this operator is where the repressor comes to bind. Remember, we said that this repressor is produced. It is just an enzyme which is produced by the lac I. Okay? So this lac I, the inducible, I means inducible. So this lac I, or the inducer, produces the repressor. Okay? Once this repressor is produced, it comes to bind at the operator. Now, when it binds at the operator, this is what happens. It will close. Okay? It comes with jaws, like it will have jaws like that. So it will have these jaws so that it closes the operator. So once it crosses this operator, this RNA polymerase can't pass. It can't pass to go and transcribe the genes we have in the structural portion. So that is one thing I want us to understand. Now, since this RNA polymerase can't pass through because there is this operator which has been produced by the inducer, the lac I, and it is sitting, or it is bound to the operator. So there is no space, there is no room for this RNA polymerase to pass. Now, what causes the repressor to get out, like to move so that this RNA to pass? What causes that? It is the production. There has to be the metabolism. Okay? There has to be metabolism. This lactose has to be converted into what? Glucose. Okay? So there has to be the lactose. There has to be this lactose being converted into glucose plus galactose. Okay? So this lactose has to be broken down into the glucose and also galactose. So when there is that metabolism, when there is that breakdown of lactose into glucose and galactose, at this point, this repressor will be turned on. okay, the repressor will be turned on. So when I say repressor will be turned on, it means that it won't bind, like it will move away, it will move a bit so that this RNA polymerase can pass. Initially, as it is right now, it has been bound. Okay, this repressor is binding to the operator. So we can say that the repressor is in an off state. So it is in an off state because this polymerase can't pass through. There is no space for this polymerase to pass through. It is binding to the operator. So this repressor is in off state. But when there is a metabolism, once it senses that there is a metabolism, when there is the breakdown of glucose into galactose, you see, there has to be lactose for this repressor for it not to bind. There has to be lactose. But when there is glucose, this repressor will still be bound. It will be in an off state, meaning that there's more glucose which can be used instead of lactose. There's more glucose which can be used in the carrying out of certain reactions. So this repressor is only turned on when there is no glucose, okay, or when there is less glucose, or in absence of glucose and in the presence of lactose. So what am I trying to say here? The point I'm trying to drive across here is that for the repressor to be turned on, there has to be presence. Okay? So this glucose has to be absent. So glucose has to be absent for this repressor to be turned on, but sucrose has to be what? Present. So when sucrose is present and glucose is absent, this repressor will be turned on. So when it is turned on, what will happen? So this is what is going to happen. So let's say this is where the repressor is. So this is where the repressor is. We have the operator there. Then we have the repressor. So when there is absence of glucose, so the repressor won't bind. So it will just be like that. Okay? It won't bind to this what? To this operator. So the repressor won't bind. What has caused this repressor not to bind? It is because there is absence of glucose, but there is presence of lactose, which is sensing that this RNA, which is on the promoter there is RNA polymerase there. So this RNA polymerase now will pass through, okay, will start to move. It will move until it passes through so that it can now transcribe the genes we have there on the structural portion. So basically this is what happens during the operation of this, what we are calling the Lac Operon. Okay? But I mentioned that there is this portion, okay, just between the Lac I there, the inducer, and this promoter. There is this portion, okay, which we are calling the cap. When you're talking about cap, you're just talking about the catabolite, okay, activation protein. The catabolite activation protein is the one now which just helps this RNA polymerase to bind efficiently to the promoter. So it helps in the binding of RNA efficiently to the what? To the promoter. And in that case, it will increase the rate at which the genes are being transcribed. Okay? It will increase the rate at which these enzymes are being transcribed. So this catabolite, as you've said, it is catabolite activation protein. It is only active when there is catabolism. No wonder you're saying catabolite activation protein. It is only active when there is catabolism. When there is catabolism, what is that catabolism? The breakdown of lactose into glucose plus what? Galactose. When there is this breakdown, when there is this catabolism, okay, that's when this cap will be active, but all this time it will just be inactive. Okay, now this activation is due to a certain what? A certain, I can say a certain substance which causes the catabolite activation protein to be active. It is only activated due to the presence of what is known as the CAMP. Okay? So we'll have what is known as the CAMP. CAMP. So maybe I can start with a small letter, then I say like that. So when you're talking about this CAMP, it is just cyclin adenosine monophosphate. So this cyclin adenosine monophosphate is the one which is going to tell the cap. It will bind to this cap, hence it will increase the rate of transcription. So this will bind to the CAMP. Okay? This CAMP will bind with the cap. Once they get bound, there will be increase in the water, in the rate of transcription. So we need to understand that. So this cap rather, it is only active when there is the presence of CAMP. So this cap activates this CAMP, the cyclin adenosine monophosphate. It is the one which activates this cap. Okay? Maybe let me write it here. Let me write it here. CAMP, you are saying it's cyclin adenosine monophosphate. So this CAMP, the cyclin adenosine monophosphate, will activate this cap so that this cap can make the RNA polymerase to bind effectively to the what? To the promoter, hence increasing the rate of what? Transcription. Now I said there is what is known as the terminator here. So this terminator here, it just causes now the deactivation. It just causes the deactivation. Like it will cause the termination. Okay? The termination of what? The processes. For example, let's say this A has caused the production of this B. Okay? Then this B has caused the production of this C. So this C will go back again to A. The production will just go back to A. Like there's a production of A. After the production of A, there's the production of B. Okay? So this is what I'm talking about. Like for example, there's a production of A. Production of A has been done. There's again production of B. Production of B has been done. There's production of C. Production of C has been done. It goes back, the process goes back to production of A again. Okay? But when this process goes back to production of A, it deactivates, like it will stop. Okay? Once it stops, then there will be no termination. There will be no continuation. So this will lead to what is known as termination. So there will be termination. So this is just the importance of the terminator. It's just like the way they operate. Okay? When we have this probe, from 0, it goes to maybe 6 hours. When it's from 6, it goes to 12. When it goes to 12, it means that the day has ended. So when it goes back to 0 again, from 0 to 0 again, like when it makes a complete revolution, it means that the day has ended. 48 hours means that the day has ended, right? So it goes from 0, when it comes again to 0, it means that the day has ended. So when it starts from 0 again, it means that that is another day. So this is the same logic which is applied also in this instance. Okay, so this is the end of the expression of gene or the creation of gene expression. This is where it ends. So what we need to understand is just the operation of the Lac Operon. Once we understand, we appreciate the operation of the Lac Operon, then it will be all well and good to go. Okay? So basically, this is what we need to understand: the operation of gene expression, the operation of the Lac Operon, the gene expression, gene regulation, the types of the genes, okay, or the classes of genes, what is involved, the enzyme produced, and things like that. So thank you for watching. Don't forget to like, subscribe, and also share so that we can move at the same pace and help one another. God bless you.